Morning, everybody. My name is James Beckley. I'm with the Virginia Department of Environmental Quality. I'm here today to talk about the Colilert procedure that the Rivanna Conservation Alliance has recently upgraded to. The Rivanna Conservation Alliance is currently level 3 status for by the Virginia Department of Environmental Quality for benthic macroinvertebrate monitoring. After today, we do the training, and then I follow up with a audit to make sure that the procedures that we're going to be covering today are being followed. RCA could become level 3 status for E. coli bacteria using the Colilert procedure. That would make RCA the first citizen group that's level 3 status for benthic macroinvertebrates and E. coli bacteria, which is a unique distinction. Level 3 status means that the data that would be collected by RCA is considered to be just as good as if it was collected by one of our staff at the Department of Environmental Quality. We use that data to assess waters for the health of waters as part of the 305B, 303D report, as well as to list and potentially delist waters from the total maximum daily load list, or the 303D list. Currently, the Rivanna Conservation Alliance is doing E. coli bacteria, but they're using the ColiScan Easy Gel procedure, which is a very good procedure. However, it's only considered level 2. It gives you a very good idea of the E. coli values that are the E. coli levels that are occurring in a water body, but they're not considered to be as accurate as a laboratory-based method. The Colilert procedure that we'll be covering today is a EPA-approved method. It's used by wastewater treatment plants, it's used by environmental laboratories, and it's used by actually our agency, the Department of Environmental Quality, for E. coli bacteria. So the bacteria bottle that's used in the Colilert procedure is a larger one than you would normally used to with the ColiScan. This is a 120 milliliter sample bottle. You'll notice here that there's a line, a little indentation right here. This is to 100 milliliter value. So under the old ColiScan procedure, you had this really small bottle because you were only pulling like 3 milliliters of sample. With Colilert, you can do a 100 milliliter sample. So you would basically fill, when we go out into the field, we'll talk about it or show it in more detail, but you would fill basically to that 100 milliliter line, and we will actually be testing that full sample volume. So you won't have to worry about any calculations or anything like that. Now, for sampling equipment wise, you have either would be collecting your sample directly in the bottle itself, like if you're wading into the stream and collecting your sample, or you would use something like a sample wand if you're from the stream bank. Some folks have also used like a sample bucket. Y'all probably use this during your ColiScan samples, where you would take the bucket, toss it out into the water or lower it from a bridge, fill it up, rinse it out, dump it, get a second sample, pull it up, and then you would pull your sample from there. For Colilert and these level 3 protocols, we really don't recommend using your bucket. And the reason why is because even if you rinse it out, you might have some carryover, some cross-contamination. Now, you can do it if need be, but we highly recommend against it. And in fact, our own field teams, what we use is we use a little bit different bucket. It's actually a stainless steel bucket. And we drilled holes on the side of it, and we ran surgical tubing through those holes. And so we have this outer loop of like this rubber surgical tubing material. And we put our bottle in that little loop to kind of hold it in place. And when we lower it into the water and collected our sample, that bottle is still outside of the bucket. So we don't have to worry about any potential contamination from inside our bucket. Now, instead of using a bucket, what we've created here is a sampling device that you're still collecting that sample directly into your bottle. And basically the way it works is you take your bottle, and you'll notice that the bottle here has it has a little plastic ring that you can remove, so it's kind of like a seal. It's a sterile bottle, just like your old ColiScan bottles are. But basically, you put your bottle in there, and then you just twist this a little bit just to kind of lock it in place. It's not going to come out. You can take off your plastic ring, or the seal, and open up your bottle. And again, you don't want to touch the inside of your bottle or the inside of the lid or anything. And then basically, if you're on the like say you're on a bridge, you would just lower it down into the water. It'll fill up, and again, it's pretty much going directly into the bottle, so you don't have to worry about any potential contamination issues. Retrieve your container, trying to get your rope in the bottle, and then cap it shut, just like that. Unscrew it just a little bit to let that bottle come out, and there's your sample. Okay, so no rinsing or anything is necessary. So we've provided some of these materials for you. These were basically made from hardware supplies, probably $2, $3 for the materials required. There would also be included a cooler for transporting your samples. You would want to have like a Sharpie marker, preferably labels to label your bottle, so you can put your information on there. You'll have a field sheet to record your field conditions, the time that you collected your sample, because the laboratory will need that. And that's pretty much it. In terms of the coolers, currently RCA has blue ice packs that they've been using. For samples like the Colilert, I believe the wet ice using regular ice cubes is better, because the blue ice packs are not very efficient at cooling down a large volume of water, say from like a 100 milliliter bottle, very quickly. It would take take several times longer than it would if it was immersed in like wet ice. So I would recommend that we go with kind of a more rigid cooler that you can put wet ice in, and you just put your bottle directly in there and you'll be fine. When you do that, because you really want to get that sample as cool down to about 4 degrees Celsius as quickly as possible to kind of stop any bacteria replication or even potentially some die-off from occurring. That's so when the lab gets the sample and processes it, that number that they're reporting is pretty much the same number as from that sample that you originally pulled from that water. So you're getting the most accurate number. This is a quantity tray. So basically, when we were going through the procedure, you would fill this with pour your 100 milliliter sample with the media mixed into it. Fill this tray, you would run it through the sealer, and then you place it in the incubator. And after incubation, or during incubation, each of these little wells is like an independent sample, okay? And so you have about 97 wells in here of different volumes. These are about approximately 1 milliliter. These are about, or maybe 2 milliliters. These are about a half of a milliliter of sample volume. And when we look underneath the comparator, we have a sample that's currently incubating right now. We'll pull that out later today. And we run it underneath the UV light, and you look at the color, it'll fluoresce, and it'll also have a yellow color. And if it possesses both of those traits, we would count that as an E. coli bacteria. And then you would use a quantity tray or a comparator chart, and based upon the number of large wells and small wells that give you a positive reaction, you get a value associated with it. Colilert media. So this is the media packs themselves. So you don't have to worry about mixing up agar or anything like that. Again, the quantity tray sealer. Then you have your incubator. This is a precision incubator. We don't recommend using anything like a chicken egg incubator, like say for example is used for Coliscan. This is a very precise incubator because you want to make sure that you're incubating your samples at 35 degrees Celsius plus or minus 0.5 degrees. So it's a very tight temperature range. Okay. And then you have basically a UV light source, and there's been a little light box, and this is basically what you're going to be using to read your samples. Move on. Let's talk about sampling safety. Just as an overview, it's very similar to what you're currently using now. It's mostly common sense. When you're out in the field, you want to be aware of your surroundings. If you are sampling, like say in a creek, and say you're going to wade down the creek, you don't want to go into a creek where the water is very high and moving very fast, because that could potentially sweep you downstream. You want to, if you're getting down into the creek or sampling from the bank, that the banks aren't too steep so you can't get out in an emergency, or there's a lot of brambles and poison ivy and everything else that would make it very painful to get in and out of the water. Most folks sample from bridges because they're very convenient to get to that middle portion of the stream. if you're sampling on a bridge, you want to make sure that you have sufficient room to sample from. So like the bridge would have a shoulder. You don't want to be sampling from a bridge where you got traffic going by and there's like one of those narrow country bridges where there's no shoulder. I've sampled those before and those are not fun. If you park your vehicle on the roadway, you want to make sure your vehicle is fully off the roadway onto the shoulder, preferably actually like in a nearby parking lot, say like around here in Charlottesville. If you're on the roadway and you're parked on the shoulder, put your flashers on. We do recommend that you always work in teams. You always have a buddy with you. They're helpful to have to help you kind of hold the lid of the bottle, kind of have an extra pair of hands if needed, but also very importantly, there's an extra pair of eyes that they can keep an eye on things and let you know. Before you go out into the field, you want to check the weather, make sure that you're not going to be, say, miles away somewhere and there's going to be some big pop-up thunderstorms and you're going to be kind of stuck out there. If it's snow and ice on the roads, it's very hazardous. You may want to reconsider sampling at another time because you don't want to put anyone in danger on that. So it's mostly just common sense. When you're out in the field and even in the laboratory, we do recommend you wear personal protection like gloves or have hand sanitizer if you're collecting samples. Lab folks, I would recommend that you have gloves all the time. That just helps eliminate any reduce cross-contamination and other issues. In the lab, it's always good to have a lab coat on. The materials that the Colilert procedure has are not overly hazardous, but you could potentially spill stuff on you, and it's just nice that if it spills on your lab coat, you can remove your lab coat and wash it and not have to worry about it. We do recommend for folks with if they're using a UV lamp to have glasses or UV goggles that have a UV coating. If you're going into the field and say you're sampling in the stream, you want to make sure you wear adequate footwear, hip waders, or sturdy shoes. Don't go into a creek at any time with open-toed sandals, or even those like the Croc type footwear, because things can poke through the holes and get into your foot, and you don't want a piece of broken glass or anything else kind of poking in that foot because you could potentially get an infection or other issues occur.
When you get out into the field, it's a good idea to fill in a field sheet. The field sheet includes important information such as the station number, which is used by the laboratory to know which sample is associated with each field sheet. In addition, the field sheet provides useful information such as current weather conditions, recent rainfall, which is important for E. coli measurements, and any other field readings that are collected while on the site. If collecting samples by wading, it's important to enter the stream downstream of where you're going to be collecting your sample and wading upstream. This is to avoid any disturbed sediment from entering your sample bottle, producing an artificial result. Here you see the volunteer scooping a sample by moving the bottle away from them towards upstream. That's so the bacteria that flows into the bottle is from the upstream source. Using a sample pole is a good idea if you're sampling along a steep bank where getting into or out of the stream could be potentially hazardous. To use a pole, you would simply attach the bottle to the holding apparatus and dip it into the stream. Again, you would have the opening facing towards the upstream portion of the stream to collect your sample. Once the bottle is full, you retrieve it and cap it and place into the cooler. When filling the bottles, make sure to try and fill to the 100 milliliter mark. When using a bridge device, simply place the bottle into the device after you remove the protective outer plastic wrapper. Lock the bottle into place and open the lid. Be sure not to place the lid face down onto a surface. Lower the device into the water body, and once filled, retrieve it. Confirm that the bottle has been filled to the proper level and add the cap. If you've overfilled, dump out any excess water. Be sure to include information on your sample bottle including the station number and any other important information, and place into the cooler.
When the samples are brought back in, it's good to put them in like a refrigerator or the designated location that is either a cooler with ice or a refrigerator. Keep your samples chilled until the technician will process the samples. For environmental samples here in Virginia, we allow up to 24 hours holding time. So if everyone brings in their samples, say on Thursday, they can drop them off, and then Friday the technician can come in and process the samples. Of course, they would have to come in the following day to read them. The first thing you would do for the technician is you would verify that you have all your field sheets and your samples and everything is properly labeled. So we have 2 samples here. One of them is not labeled though, but for today they were collected from the same site, but we do recommend labeling your bottles. The technician will also grab the Colilert media that they will need, and also the quantity trays that are going to be necessary. We're going to process 2 samples today. Now, it's important for the technician to wipe down the area that they're going to be working on. Using isopropyl alcohol is probably the best substance to use. I do not recommend using bleach, because bleach has the residue can actually contaminate your samples because it leaves a little bit of a film afterwards. The alcohol, the nice thing is that it'll pretty much evaporate. I also don't really recommend using Lysol wipes. The technician should turn on the tray sealer right here, and it'll take a couple minutes for us to warm up. The yellow light or the orange light that you see right here, it means that the tray sealer is warming up. It's not ready to process a sample. When it turns green, then you can run your tray through that sample. Okay. Next thing the technician will do is label the quantity trays. Now the technician is going to be recording some information here on their laboratory sheet. Now the sheet that was submitted by the volunteers is the field sheet, but there will be most likely a secondary sheet for the technician to process lab specific numbers. And so one of the things that the technician is going to record is they're going to record the lot number and the expiration date that's printed on the end of this box of the Colilert on their log sheet. They're going to record the samples that are going to be processed during this run, and the amount volumes that are going to be tested. Generally it's going to be 100 milliliters, and the date and time that those samples were collected, so they're going to record that off of the field sheet. Okay. When the technician receives the bottle, they should look at the samples and verify that they're within the 100 ml line and the between the 100 ml line and the bottom of the shoulder of the bottle. And if the bottles are overfilled, and you should do it before you put the media in there, you would use a sterile pipette to withdraw any excess water, or if it's below the 100 milliliter line, you can use a sterile water to bring it up to that 100 milliliter volume, but you got to record on your lab sheet the actual volume. So if you added, say, 6 milliliters to it, your sample volume is now 92 or 94 milliliters. Okay. So we have our one sample, and this is going to be our second one right here. And so we're going to take the Colilert media, and they come in these little snap packs right here. And what you want to do is you just want to shake the pack to kind of get everything settled on the bottom, just like you would for, like, say, ketchup or whatever. Just make sure it's all settled down in there. And then, while they're wearing gloves and everything, they're going to snap open the pack and open up the bottle. And it's always good to open one bottle at a time. Don't have multiple bottles open, because then you might cross-contaminate your lids. Dump the contents in there. Get as much in there as you can. And then close the bottle. And you can process, if you got them lined up like this, for example, you can process multiple samples at one time. So we're going to do this for the second one right here. And then what you're going to do is you'll notice here that the bottoms of the bottles, you got a little bit of media. It's kind of still like a little powder in the bottom. So you just want to mix them up. You can do them one at a time, or if you're fairly skilled, you could do both. Don't do more than two. But you just want to swirl them and kind of mix them up. So once the samples are, or once the media has dissolved, you'll see here, and of course it's condensating in here, but there's no more powder in the bottom. And the water's taken on kind of a little bit of a yellowish tinge to it, okay? So by about the time that you're done getting your samples prepared, your tray sealer should be about up and running. The light's green, and you can also hear the cooling fan running, all right? So now we're going to fill up the quanta tray, all right? And so the tray is kind of kept closed, and it's a sterile tray. So when we are accessing it, you don't want to stick anything in there, right? So what we recommend doing is one bottle at a time. Just kind of loosen that bottle lid to just let it just sit on top. And then you kind of, with the plastic side facing your palm, you peel, you kind of bend it, bow it inwards, and then you kind of pull this flat back a little bit, and that'll kind of create a little pocket, just like that. Okay. And then what you do is you pour your sample volume, and you really don't want to spill any of this, because then you won't be having 100 milliliters. So get as much of that out as you can. And then you close your pack. Now, you'll notice here that there's some bubbles right here, and that sometimes happens, and that's okay. What you can do is just kind of tilt it at an angle and just give it a little bit of a tap to dislodge the majority of those bubbles. Okay, you want to try to, it's okay there's a few bubbles left in there after we seal it, that's fine, but you want to get rid of any big bubbles. Okay. Then what happens is you take this tray sealer, and you'll notice it has the same indentations as the Quanta tray, so this is the Quanta Tray 2000 sealer tray. And you basically just lay that tray in there until it fits in there. So once you got your tray prepared, you place it in this input tray right here, and you just push it in until it grabs it, and it's going to run it through the sealer. And you want to have your hand on the other side to get it. And you pull it out after it's, and now the sample is sealed. See? So now if we look at our tray, this is what I would call a very good tray. There may be a couple small bubbles in there, but that's okay. As long as the bubbles aren't more than about halfway full on those pockets, you're fine. So this is a good tray. So what we will do now is you can process your second sample or your subsequent samples. Once you've have about say 10 samples worth, or you're done with all your samples, you want to load them into the incubator. Again, we don't recommend you open the incubator door every time to put every individual sample in there, because that incubator will start losing its temperature. But once you've gotten your samples that you're ready to process, then we're done with the sealer, so you can turn it off. What you want to do is you want to record the time that they're going to go into the incubator. Okay, so we're going to record the time here. That is when we place in the incubator. And you're going to record that same time on the log sheet, where it would say on the space here of placed date time incubated. Now the other thing a technician will do is every day that the incubator is either going to be used to incubate samples, or when you're pulling the samples out, you want to record the time, or you want to record the temperature of that incubator. So it's recommended that you do this before you start putting your samples in. So currently we're reading 35.1 degrees Celsius. Again, you're allowed 35 plus or minus 0.5. So it's 35.5 to 34.5. So you record it on your little log sheet here, the date and the time, or the temperature, which is 35.1 today, and the time that it was placed in the incubator, and then the technician's initials. You don't need to record the time when you're not incubating samples. It's either the day that you're putting samples in, or the day, or the following day, and the following day that you're taking them out. The rest of the time, the incubator could either be turned off, but you don't need to worry about the temperature. Now, we do recommend that you do not rely solely on the provided temperature on the incubator, because over time, incubators, the thermistors that are in here can get inaccurate. So what we recommend is have a small thermometer, a secondary thermometer that's kind of in a liquid medium, like a gel, like a glycol, and confirm that that temperature reading is in the same range as your incubator. So I'm just going to pull it out because, again, it's in that glycol, so it holds that temperature longer, but we're right at 35 degrees. Okay. So now you take your samples and you would load them in your incubator, and you'd place them upside down, so the plastic is facing down. It doesn't quite matter if it is facing up, but we found that facing down, at least you have all your informations on the back, so you're not grabbing the wrong samples when you're retrieving them. And the trays that are set up here in the incubator are set at the right locations. You really want to avoid, well, this top tray can move up actually another rack. You do want to avoid not putting your trays resting on the very bottom of the incubator. Like where this bottom rack is, that's as low as you want to go. And you don't want to have trays really within about 6 inches of the top part, because there is a little bit of temperature differences, because that's where the heating coils are. All right. So that is all that is needed for the loading your samples. All right. So now the samples are going to be incubated. The ideal time is 24 hours. However, you're given, I believe, 2 hours. So 22 to 26 hours is the optimal time to pull them out. If you pull them out a little bit earlier than that or a little bit later than that, it's probably going to be okay, but you really do want to try and stay within 22 to 26 hours incubation. All right. If you let them sit there much longer, after 26 hours, the compound that causes the bacteria to fluoresce will start to break down, and so it'll get harder and harder to identify the bacteria. Okay. So we're now 24 hours into the future. Just like a cooking show, I have prepared my turkey.
Julia Child.
I'm Julian Childs in this training video. So I came here yesterday and we processed a sample from the same site. So we're going to retrieve that. I've already recorded the temperature of the incubator before I pulled that sample out. So this is your Colilert sample. I knew it was going to be high because it had rained just the following day. See that nice bright yellow color? That's telling me there's coliform. Now, is every single well yellow? Looks like it.
Yeah.
Large and small. So we already know that we're at the maximum detection limit, which is over 2,400 coliform. We already know that. Now let's put it underneath the UV light and let's see what happens. What you would do is you turn on your UV light source, and it's on. And then you place your tray in the light box here, and I'm not surprised by the result I'm seeing. And you look through, and then you start counting the number of positive wells. So we can have you come in, and if you want to take a look, and you can look in there and see how it fluoresces. It's a very strong fluorescent color. Colilert does provide basically a check. So if some folks are like, well, is that a a fluorescent sample. They provided a sample where it's basically the minimum glow that you're looking for. So we're going to take our sample out, and we're going to put this one in. And you'll notice here, very different color, right? But this is, if you put this underneath UV, you can still see that strong fluorescent color. It's not as bright as the current sample, but that's okay. So the tray that they provide you kind of gives you a nice baseline of this is the minimum glow that you want to look at. If it's any brighter than that, it's definitely E. coli. If it's any less, maybe not so much. Okay. Now let's say that we don't have all of our cells are positive, right? Well, how do we calculate our bacteria numbers? This is where you would use the provided Quantitray 2000 MPN table. Okay. What this table does is on the top row is the number of small wells, and on the side is large wells, and you basically use it like a grid and match it up. Okay. So for example, for today, well, they're all positive, right? So we go onto the back, it's a double-sided, and we have 48 small wells. And there's also 49 large wells. And we're now greater than 2,419.6 E. coli. That's the MPN value if everything is positive. And that's another nice thing about Colilert is that you're able to get up to over 2,400 bacteria numbers using a 100 milliliter sample. You don't have to subdilute, which always it gets be a little problematic because you're like, did that smaller sample I pull out, is that still representative of the number? Now, if for some reason you have to dilute, because say you got a bottle and it was like say 3/4 of the way full and you had to add 20 milliliters, that's going to affect your MPN numbers. You're going to have to do a little bit of calculation to say, okay, if I did 97 milliliters of sample, I added 3 mils of blank water, that's going to, I got to use, do a calculation to say, well, it's going to be greater than 2,416 or 19 colonies, 0.6. It's going to be like 2,600, 2,700 bacteria, based upon how much you've had to add of bacteria-free water. But most of the time, you shouldn't be doing that anyways. You'll just process a 100 ml sample. You'll notice here on this particular tray, this cell right here is about halfway full. So it's got a bit of a larger bubble, more notable size bubble. So that's, if you're any less than that, then you may want to not count that well. All right. So again, if you did the shake your Quantitray before you run it through the sealer, it should eliminate those bubble issues. But if you have one that's like half full or less than half full, it's very difficult to determine if that color that you're seeing is due to the bacteria was present or if it's just because you have such a smaller volume that you're dealing with. So just be careful when you're loading when you're sealing the tray. If you do get one that's kind of empty, just don't count it. It's not going to really affect your overall results that much. It really isn't. Disposal. If you have the equipment, some places talk about putting these in an autoclave and autoclaving it to kill the bacteria in here. However, since these are sealed, they're pretty well protected from potential contamination issues. So they could be disposed of in the trash if you don't have a mechanism to sterilize, like an autoclave. We do not recommend you try to pull these open and drain the stuff out and rinse it. No, we're more likely to cause problems than you're going to solve. So you can just dispose of these in trash. If you have the ability for, like, say, biohazard type bags and that type of stuff, we recommend you do that. If you got that type of pickup, try to avoid putting it in general trash. But it depends on what your resources allow for that. But generally speaking, most of the stuff that's growing in here is E. coli or coliform bacteria. There should not be as much of a risk of pathogens as maybe some other methods, but there's still that potential chance. Your sample bottles that you had used the previous day, we recommend you just rinse them out. They can go straight in the trash. Do not reuse the sample bottles. Do not reuse any pipettes. It's single-use stuff, okay? because again, you can't verify if that cleaning was sufficient. And since we're now talking about quality assurance, let's talk about some of the other things. For your field staff, or for folks that are sampling in the field, we do recommend that if, say, for example, you're doing a 15 sample run between the various volunteers, and they're collecting, say, 15 samples worth of stuff on one day, you should select one of those persons to do a blank and a replicate sample. Okay, a blank sample, that's just to verify that your bottles are sterile, and it also verifies that any equipment that's coming into contact with your sample is also clean. And it also checks to see if the technician or the field person is using good protocol. You know, they're wearing gloves and what have you, and they are not adding bacteria due to handling the bottle like they normally would, you know, while they're processing or collecting their sample. We also recommend that you do a replicate sample, and we generally recommend you do that with the same person that's doing the blank, just because that's so you have them all together and that you can fully test the proficiency of that person all across the sampling protocol. And basically what a replicate sample is that you give them an additional bottle. Like if we had like one of those bridge sampling devices, they put the bottle in, lower it into the water, collect the first bottle, pull it up, cap it, put the second bottle in, and put it back down immediately to collect that second sample. And what you want to do is you just want to, that replicate will determine if you have any variation in your samples. And you know, if you collect them at the same time or very quickly one after the other, it's unlikely that the change that you're seeing is due to the environment change. It may be due more to a technique issue. And you would just process them, and then you would label those bottles as like, you know, the station ID and then like 1, station ID 2 to let you know that that's your replicate sample. Now in the laboratory, there is some quality assurance that the laboratory person should do to test their technique. Like say if you're testing or if you're having to use spring water, I would do it blank to verify that your water is still pure, it's bacteria free. There is also a test that should be done for each batch of, or each lot of Colilert media. The IDEX and we recommend that you run a quanti-cult test. The way the quanti-cult test works is you have in here 3 sets of vials. One is E. coli, one is Klebsiella pseudomonas, and the other one is Pseudomonas aeruginosa. Thank you. So E. coli, you should be seeing that yellow color and it should fluoresce. The Klebsiella is a coliform bacteria, so you should only see the yellow color, no fluoresce. And the Pseudomonas, you should see no color and no fluorescence. Okay. And what this does is this ensures that the media you're using isn't contaminated, didn't come out from the manufacturer contaminated, or that your technique didn't cause contamination to occur. Okay. And the way the procedure works out is you take each vial, E. coli, Pseudomonas, and Klebsiella, warm it up in the incubator for a few minutes, about 10 minutes or so, just because it came out of the freezer. And then dump that contents of that vial into a bottle filled with, you know, your bacteria-free water, and then process it as a normal sample. And then the bacteria concentrations in here are of a known quantity, and that's again to verify the technician's technique, and also it also checks to make sure there's no contamination with the water you're using, as well as the media that you've purchased.
How many vials are a lot of the media?
The Colilert, I mean, they make them into like huge batches. So I think all the batches that RCA has purchased, there's about 200 samples worth. I think they all came from the same lot. So, you know, you could do this one sample at a time, but since you have 3 of these and they have a 6-month or so shelf life, it wouldn't hurt to run these even through the same lot periodically at least I would say recommend it at least twice a year to test that technician's person, or if you're bringing a new person on board, you can use this as a form of like a proficiency check for them. Okay, so that's about it for the quality assurance for all of these for your laboratory side of things. Again, because you have the incubator and we have a secondary thermometer in here that takes care of all temperature issues. We don't have to autoclave anything, so we don't have to check our autoclave to see if it's properly sterilizing anything. So it's very minimum of what's required to confirm that you got a good sample. So that's pretty much it.